Hi guys, welcome to class. So I promise that it's not the bacteria that causes human disease. The other guys in that equation, that is virus, protozoa, fungi, they also cause diseases as well. So let's consider viral infections. So now, when we're talking about cholera, where we said the causative agent is Vibrio cholerae, and I said it's one of the most deadliest diseases, and I said the hallmark of cholera is massive dehydration, because when the Vibrio cholerae gets into the system, it gets to the GI, stays around the lining of the GI, the intestine, and it releases this toxin that will induce massive loss of water, resulting in the massive dehydration. You see, for virus, before virus can cause any disruption in the normal functioning of the body system, it needs to get into a whole cell. It requires entry for it to cause disease. It requires entry to the genome. Into the whole cell. Now, what do you mean by genome? And by genome, we're talking about the nucleic material. The nucleic material. So by genome, we're referring to what? The nucleic material. Are you with me? Yes, you should. By genome, we are referring to nucleic material, and there are different means in which it gets in. The one is by direct fusion, in which particularly those viruses that are enveloped, so they just fuse directly with the cell membrane and they just enter. While in the case of bacteriophage, they use a kind of a probe to insert their genetic material into the bacteria. Do you understand? So that this virus can infect bacteria as well. So there are different means. Some is through receptor, in which they communicate with the receptor that will not create a kind of pore or a kind of channel through which they can come in. The idea is the genome has to be entered. There has to be entry of genome into the host. And even though it comes in as the O together with the capsid and the rest, there has to be an uncoating to reveal the genome. Because it's this genome that will not that will get into the nucleus of the cell. In the case of a DNA virus, remember I told you when we talk about virus that the genetic material can either be DNA or RNA, but never both. So it's now going to, if it's going to be a DNA virus, to get to the nucleus of the cell, then use the machinery of the cell to replicate itself, form its mRNA, then translate. But if it's going to be an RNA virus, it's going to happen within the cytoplasm, because the cell is going to recognize the RNA of that virus as the cell's virus, then translate it, forming proteins. These proteins, it will then arrange it, couple it, make up its capsid codes, as well as any other protein in it, and to leave the cell. Now, there's also something unique about how viruses leave the cell. Now, a virus can leave the cell by cell lysis or by exocytosis. So, this is also very key in understanding virus. So, exit from the host cell. So exit from the host cell can be via cell lysis or budding. That budding, they also refer it to as exocytosis, that is the cell vomiting. Because now, in a case of viruses that are naked, they are going to leave the cell by cell lysis. They have to destroy the cell for them to find their way out. But if the virus is enveloped, it will just bud out, because it has an envelope. It just buds out of the cell, and that thing is very key, because there's something we call latency in virus. Because most sexually transmitted diseases like gonorrhea or chlamydia, most of the time, particularly gonorrhea, they don't present. Even syphilis, they don't present with symptoms. Why? Because most of the viruses like Wucheria, gonorrhea is enveloped, so it will just be reproducing, as well as HIV, even HIV virus. it will be reproducing within the cell. Because it's envelope, it can easily get out without causing lysing of the cell. So it would have, and that's the reason why by the time HIV finally presents itself, the person will present with so much symptoms and the person will be down, because the virus has multiplied enough to cause catastrophic effects, and the person is unaware because it's not causing lysis of the cell. But in a situation where it causes lysis of the cell, you are going to feel it that, like hepatitis, you are going to feel it that, yes, you start presenting with symptoms. And that's the same thing with herpes virus. Herpes virus to displace that. That's the reason why, particularly sexually transmitted disease, you don't know until you start to present with symptoms. So the symptoms doesn't start immediately. And when the symptoms start, it's going to be very severe because this virus would have sufficiently multiplied itself. And that's why when people present with HIV virus, they have so much viral load that the goal of management is to try to reduce the viral load. What I'm just trying to say is that before the virus can cause any infection or cause an illness or a disease, it has to get into the whole cell. That's the difference about virus. Bacteria doesn't need. Some bacteria, like Rickettsia, they are intracellular pathogens. Even Mycobacteria tuberculosis is regarded as an intracellular pathogen, intracellular bacteria. But most times, bacteria can stay within the interstitium. They don't need to get into the cells, and yet they will cause the disruption they want to cause. But for viruses, they need to get into the whole cell. That's one classic difference between a bacterial infection and a viral infection. And the thing is, with viral infections, you do not actually kill the virus. You understand? What you just do is that you just try to limit the growth of the virus. When bacterial infection, the antibiotics that we say they are bactericidal, they can kill the bacteria. Some are bacteriostatic, they can limit the growth of the bacteria by inhibiting the protein synthesis. But for antiviral drugs, you just want to manage the reproduction. You don't want the reproduction to be excessive. You want to control it, you want to reduce it, but you can't succeed in killing it. That's why viral infections are much more severe, like the HIV, are much more deadly than bacterial infections. So I'll see you in the next class. We'll talk more about viral infections.